Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing GAT B question paper conducted in the year 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question paper of GAT B being discussed, you can go to my channel Omni Bioentrance. In the playlist you can see for GAT B, in there you will find. Okay. For an imaginary Martian species. Martian species means the one which is found in Mars. Okay, it is imaginary. With three nucleotides X, Y and Z, how many three-letter codons are possible? So for Earth species, we have got 4 nucleotides and 3-letter codons. So how do we calculate? So 4 raised to 3, that is 64. So here it is 3 nucleotides. So 3 nucleotides X, Y and Z. How many 3-letter codons are possible means 3 raised to 3, that is 27. So the answer here is option 2, 27. What is the likely number of amino acids in a 11 kDa protein? So we know the molecular weight of amino acid is 110 Dalton, that is 1 amino acid is 110 Daltons. So 11 kilodalton is how many amino acids will be there? So 11 kDa divided by 110 Dalton. So that will be 100 amino acids. So the answer here is option 2. For a normal Gaussian distribution. So this is the normal Gaussian distribution, this red line. Okay, that is the normal Gaussian distribution. These are the standard deviations from the mean, and this is the mean. Okay, decreasing the spread. So here you decrease this red line. That is, you do like this. This is decrease in the spread and increasing the height. So height has been increased. Height has been increased. Spread has been decreased. Would lead to smaller value of standard deviation. Yes. That is correct. So first option is correct answer. So here standard deviation is reducing. When you decrease the spread, standard deviation is reducing. The mean, now they have given options on mean also, but mean of the distribution would not change as the distribution is symmetric about the mean. Okay, so that is not going to change. Only standard deviation will change. So it will result in smaller value of standard deviation. Then next one. Which of the following is a method to conduct phylogeny of proteins and DNA sequences? It is a maximum likelihood. Okay. Then next one. The degree of inhibition for an enzyme catalyzed reaction at a particular inhibitor concentration is independent of the initial substrate concentration. That is what kind of inhibition it is. Okay, that is what they are asking. So uncompetitive inhibition, the inhibitor bind to the enzyme substrate complex. So it is going to depend on the substrate concentration. Then non-competitive inhibition, it binds to the region other than the site where the substrate is binding without affecting the substrate binding. So it is independent of initial substrate concentration. So 2 is the answer. Competitive inhibitor, it competes with the substrate for the binding site. So that's also not correct. And in mixed inhibition, it is a combination of competitive and non-competitive inhibition, and it binds to both the active site and the enzyme substrate complex. So Second is the answer. An enzymatic reaction exhibits Michaelis-Menten kinetics. What will happen if the concentration of enzyme is doubled? So keeping concentration of substrate greater than enzyme concentration or much higher than the enzyme concentration, that is as per Michaelis-Menten model. So here enzyme concentration is doubled means the Vmax will increase because it is dependent on the enzyme concentration. Because more enzyme molecules will be available to catalyze the reaction. But Km, it will remain the same. Because Km measures the binding affinity of the enzyme for the substrate. So it is not affected by enzyme concentration. So Vmax will increase but Km will remain the same. Answer is 3. A good resolution in ion exchange chromatography is obtained when the two proteins have. So there should be large difference in binding affinity and small dispersion. So large difference in binding affinity means one protein will bind more strongly to the ion exchange resin than the other. So it will help in better separation. And small dispersion. That means the protein bands remain narrow and distinct, allowing for better resolution. So answer is option 3. Given below are two statements. One is labeled assertion, other is reason. Assertion. Bacterial liposaccharides on its own does not induce memory B cell in humans. Yes, that is correct. Because liposaccharides do not activate T cells. So as a result, they cannot provide the necessary T cell help to generate memory B cells. So lipopolysaccharides on their own will not induce memory B cells in humans. Okay, so both A and R are correct, and R is the correct explanation of A. The allergic immune response is characterized by the increased level of. So it is IgE. It is allergic response. In response to allergy, it is IgE that will be increased. Then IgA, it is involved in mucosal immunity. Then IgG, it is the abundant antibody and it is involved in various immune responses. IgM is the first antibody to be produced in response to a infection, that is primary infection. Okay, then next one. The presence of antibody in an infected patient's serum can be detected by, of course, it is Western blot. ELISpot, it is used to quantify the number of cells that produce a cytokine. That is ELISpot. PCR is for amplifying DNA sequences. Northern blot is to detect RNA molecules, not antibodies. So fourth is the answer. What will happen to immune cell development if we remove the thymus from the neonatal mice? So thymus, it is involved in the development and maturation of T cells. So without thymus, T cell maturation will not take place. So T cell maturation will be impaired. So answer is 3. Not B cell maturation, because B cell maturation occurs in the bone marrow and it has nothing to do with thymus. Then a series of spontaneous point mutation occurring gradually resulting in change in influenza virus surface antigen over time is called antigenic drift. It is not shift, because antigenic shift it result from genetic, I can say, reassortment between two influenza viruses. That is, one is derived from animal, other is of human origin. If they undergo genetic reassortment, then it can give rise to a new influenza virus which will infect human beings, and it will result in a pandemic. Okay. So that is the reason for antigenic shift. It is not a gradual change, it is spontaneous. So that is antigenic shift. So the answer here is gradually resulting in change in influenza virus surface antigens over time is antigenic drift. Then next one, myasthenia gravis is an autoimmune disease where patient makes antibodies for its own acetylcholine receptor protein, which is found at the neuromuscular junction, and disrupting the acetylcholine receptor protein will result in disruption of transmission of nerve impulses to muscles, and this will result in muscle weakness, fatigue, etc. Then node 1 protein is involved in the recognition of bacterial peptidoglycan and thereby activate the immune response. TLR11, that is toll-like receptor, it recognizes bacterial flagellin and activate immune response. RIG1, that is retinoic acid inducible gene 1, it is a protein that recognizes viral RNA and activate immune response. Then next one, toll-like receptor 9, recognition of unmethylated CpG dinucleotides, that's correct. T helper cells, recognition of antigen with MHC2 complex B2. T cytotoxic cells, recognition of antigen with MHC class 1 complex, yes. Plasma cytoid dendritic cells, type 1 interferon production. So A1, B2, C3 and D4 is coming for option 4. So answer here is option 4. Then what is the length of peptides binding to major histocompatibility complex class 2 molecule? So 8 to 11 amino acid is for class 1 molecule, MHC molecules. So 21 to 27 is not MHC class 2. 15 to 20 amino acids, yes, that is MHC class 2. And MHC class 2, these peptides, they will be processed and presented to CD4 plus T cells on antigen presenting cells. Then next one, tuberculosis is caused by Mycobacterium tuberculosis. The TB vaccine is made using Mycobacterium bovis, and it is closely related to Mycobacterium tuberculosis. Okay, so second is the answer. CD4 antigen is absent on, so it is not present on which cells? So it is present in T cells, macrophage, gamma delta T cells, but it is not present on B cells. They do not express CD4. So the answer here is option 1. Then next one, HIV. HIV has got reverse transcriptase, so A4. Okay, 2 and 4 we have to see, 1 and 3 we will not take into consideration. Influenza virus. Influenza virus is segmented RNA genome, so B3. Hepatitis C virus is RNA dependent RNA polymerase, and pox virus is double stranded DNA virus. So the answer here is option 4, that is A4, B3, C1 and D2. Then hepatitis B virus genome is, it is partially double stranded DNA. So it is a circular DNA molecule, and one strand is complete while the other strand is incomplete and has a short single stranded region. That is why it is partially double stranded DNA. And then next one, herpes simplex virus maintains latency in, it maintains latency in the neuronal cells. So during latency the virus remains dormant and it does not replicate. Okay, so that is latency. So it maintains latency in neuronal cells. Pox virus replicates in, so pox virus, it is a unique in DNA viruses, it replicates in the cytoplasm without entering the nucleus. So answer is 1. Then next one, which of the following viruses is plus sense single stranded RNA virus? So it is dengue virus. Rotavirus is double stranded RNA virus. Adenovirus is a double stranded DNA virus, and influenza virus is negative sense single stranded RNA. So this plus sense single stranded RNA virus, they can be directly translated into proteins. Okay, that is why they are plus sense or positive sense single stranded RNA virus. Then next one, in N-linked glycoproteins, carbohydrate moiety is attached to which of the following amino acid? It is asparagine. Asparagine, and this occurs in the endoplasmic reticulum. Then the catalytic triad of chymotrypsin is composed of aspartic acid, serine and histidine. Answer is option 1. Then next one, a mixture of proteins A, B, C, D is separated on a Cephedex G200 column. So Cephedex G100 column means it will separate based on molecular size or molecular weight. The proteins elute in the order A, B, C, D. Assuming that all proteins are globular and monomeric, the protein with minimum electrophoretic mobility on SDS page. So here these proteins, they are loaded onto Cephedex G200 column. They elute in the order A, B, C, D. It indicates that protein A is the largest, because in Cephedex column the largest protein it will elute first, the smallest is going to elute last. So the first one, that is A, is the largest protein and it will elute first. So D here is the smallest protein and it is having low molecular weight and it will elute last. Now what is the question? The protein with minimum electrophoretic mobility on SDS page. So SDS page, it separates protein based on their molecular weight. So the smaller the protein size, it will migrate faster in the gel. Okay. But larger the protein size, it migrates slower. So here I told you protein A is the largest size, or I can say it is having the largest molecular weight. So it will have minimum electrophoretic mobility on SDS page. So the answer here is option 1. Then next one, beta oxidation. Okay, beta oxidation uses thiolase enzyme for its reaction, oxidation reaction. So A4, okay, that is there in option 3 only. Then glycolysis, it uses phosphofructokinase 1. Gluconeogenesis, it uses phosphoenol pyruvate carboxylase. Calvin cycle, it uses ribulose biphosphate carboxylase. So the answer here is A4, B2, C3 and D1. Then lipid raft are composed of. So what are these? Lipid raft. They are small and dynamic region of cell membrane, and they are made of proteins and lipids. Okay, that is lipid raft. So they are composed of. So what are they composed of? They are composed of sphingolipid and cholesterol. So the answer here is option 3. Then next one. List of one is of pollutants and list 2 is impact on the environment. So carbon monoxide, it is going to cause greenhouse effect A1. A1 is there in 4 only. So that must be the answer. Hydrocarbons, it is for photochemical smoke. Oxides of nitrogen can cause acid rain. Ozone near the Earth's surface impaired plant growth. So A1, B2, C3 and D4. Then the presence of excess nutrients in aquatic system will lead to crustacean bloom. No, it is algal bloom. So excess nutrients, it will result in overgrowth of algae, and that can cause algal bloom. Then which of the following is commonly used for converting cellulose raw material into glucose? So it is Trichoderma viridae. It breaks down cellulose into glucose. Saccharomyces cerevisiae, it is used for fermentation, not for cellulose conversion. Acinetobacter radioresistens, it is also not used for cellulose conversion. Bacillus amyloliquefaciens, it break down starch, not cellulose. Then next one, the biocide DDT, a chlorinated hydrocarbon, has a half-life of around 2 to 15 years. So second is the answer.